This is a GCSE Biology presentation on monoclonal antibodies. To get started, on our previous presentation we looked at drug trials. There are 5 terminologies that you need to be able to define. Pause the presentation, work out your definitions, and when you're ready you can restart the presentation and check your answers. Pause the presentation now. So a double blind is where both the doctor and the patient do not know who's been given the active drug and who's been given the placebo in any experiment. Remember, a placebo is a fake drug that looks exactly like the real drug, but it has no active ingredient. Toxicity refers to how severe the side effects of a drug are. Efficacy is about how good a drug is at treating a particular disease. And dose is about how much of the active ingredient a drug contains in order to be effective. In this presentation, we're going to look at define the term monoclonal antibodies, and we're going to go through the actual process of producing monoclonal antibodies. So a monoclonal antibody, the term comes from mono meaning one and clonal meaning clone. And monoclonal antibodies is where you have multiple antibodies that are exactly the same in terms of the structure. It's where you have the antibody is complementary to the same antigen, and all of the antibodies, because they have the same structure, are complementary to the same antigen, and each of the antibodies is specific to a particular antigen. Remember, antibodies are of course produced by white blood cells known as lymphocytes. When we talk about making monoclonal antibodies, we mean in terms of industrial level making them. We don't mean in terms of naturally occurring within the body. If you're looking for naturally occurring antibodies, then you need to go and look back at the presentations on the immune system. Look at the picture, pause the presentation, and have a guess of what you think the picture represents in terms of steps for making monoclonal antibodies. So pause the presentation now. So step 1 is where we have to introduce often a vaccine, but it's got an antigen on it and we're injecting it into a test mouse. In step 2, again pause, when you're ready, restart. In step 2, what happens is that the mouse will naturally produce the lymphocytes. Remember, lymphocytes are white blood cells, in exactly the same way as any animal produces lymphocytes in response to an antigen. Once again, pause the presentation, make your best guess, and when you're ready, restart the presentation. In step 3, what we have is we have the white blood cells, the lymphocytes, producing antibodies inside the mouse, and those antibodies are specific to the antigen in the vaccine that the mouse has been injected with. In step 4, again pause the presentation and restart when you're ready to check your answer. We take away part of the spleen, the groups of cells from the spleen. The reason we take them out of the mouse is because the spleen is responsible for the production of lymphocytes. And by removing a small number of cells, spleen cells, we can start to produce large numbers of lymphocytes. In step 5, we have a number of new terms that you probably have never heard from. Firstly, we have what we call as a melanoma, and a melanoma is a white blood cell that is cancerous. And we take that melanoma and we fuse it with the spleen cells that we've taken from the mouse. When we do that, what we end up with is what is called a hybridoma. And a hybridoma is a cell that's able to multiply indefinitely, and because it contains the spleen cell from the mouse, it's also producing large numbers of antibodies. Pause the presentation, work out step 6, and restart when you're ready. So in step 6, what we have is we have a number of these hybridoma cells. They're producing lots and lots of identical copies of each other and producing lots and lots of antibodies. Remember that those antibodies are specific to the antigen that we injected the mouse with at the start, and they need to be collected and purified, ready for use. So this is a summary of the steps. You need to make sure you learn these off by heart. This is GCSE Biology only content on the AQA course. Therefore, if you are doing the GCSE Biology, it is highly likely that you'll get some sort of question on monoclonal antibodies or the uses of monoclonal antibodies, which will be covered in another presentation. One of the questions that they may ask you in the exam is about evaluating uses of monoclonal antibodies. So basically, you got to come up with reasons why using monoclonal antibodies is good or bad. Pause the presentation, try and come up with your best answer, and when you're ready, you can restart the presentation. So pause now. So these are just some general ideas of statements you can use when evaluating monoclonal antibodies. Some of them are more specific to different types of treatments than others. But the obvious ones to go with are a case of monoclonal antibodies can be used for a lot of different things, including targeting cancer cells and pregnancy tests, which we'll look at when we go through the uses of monoclonal antibodies. We can produce large numbers of them very quickly. Downsides are that they do have side effects. They do have quite serious side effects in some cases. The drug trials can go wrong. They can be expensive to produce, and some people cannot have the treatment because the antibodies are foreign substances and it can clash with their particular religious views. The last one, probably the most common one, is the objection in terms of the use of animals and testing on animals. Now, it is worth noting that the mouse, when you have the small surgery to remove the spleen cells, you are not removing the entire spleen, therefore the mouse can survive. However, you are still testing on animals, and obviously some people have some strong ethical views on that. To finish off, should we be allowed to continue to use monoclonal antibodies at the expense of animals? There's no right or wrong answer to this. Just come up with some of your own personal views. Again, if you can come up with some extra research papers that support your points of view, that is even better.